technological advancements we've witnessed over just the last few decades. I mean, we've mapped the entire human genome, we're literally editing DNA with CRISPR, and we have these incredibly targeted immunotherapies for cancer. But when you look at the most widely used drugs in psychiatry, the standard antidepressants and antipsychotics sitting on pharmacy shelves today, the prototypes for their basic mechanisms were actually discovered all the way back in the 1950s. And honestly, that is the shocking reality of mental health treatment right now. As former NIMH director Stephen Hyman bluntly pointed out, drug discovery in psychiatry has largely been stagnant. It's kind of the central mystery, and frankly, the central crisis of our modern medical era. We are facing a massive global health burden when it comes to psychiatric disorders, yet we have this painfully slow, impossibly expensive pipeline for creating new treatments from scratch. So welcome to this explainer. Today, we are diving into a revolutionary framework that might just bypass this entire pharmaceutical bottleneck. We're going to explore how computational biology is acting almost like a forensic detective, matching complex psychiatric diseases with unexpected, already existing cures. And look, we aren't just speculating here, we're breaking down a rigorously peer-reviewed investigation led by Han Chong-so and a phenomenal team of researchers, which was published in the highly prestigious journal Nature Neuroscience. They've essentially built a mathematical matchmaking system to crack the psychiatric drug code, and the results are absolutely fascinating. Section 1, the psychiatric drug crisis, a stagnant pipeline. Psychiatric disorders inflict an unbelievable burden on global health. Yet our treatment strategies are, well, they're far from perfect. Conventional drug development is notoriously expensive and it takes forever. But psychiatry has a very unique hurdle. These are highly complex diseases. Schizophrenia or bipolar disorder, they aren't caused by just a single broken gene that you can easily target with one chemical. They involve this massive, intricate interplay of multiple genetic and environmental factors. For decades, the industry has relied way too heavily on tweaking those old 1950s prototypes, drugs like imipramine and chlorpromazine. The researchers behind our paper realized that trying to build a single magic bullet from scratch was just a failing strategy. Section 2, a genomic matchmaker, the hypothesis-free approach. So if building from scratch is too hard, what's the alternative? Well, the researchers turned to a clever strategy called drug repositioning. This is the process of finding new uses for existing, already approved drugs. Think of it as the ultimate recycling program for medicine. If you can prove an existing drug works for a new disease, you bypass years, sometimes decades, of expensive safety testing simply because the FDA already knows the drug is safe for human consumption. It drastically hastens the whole development cycle. But here's the catch. How do you know which existing drug to recycle? The researchers devised this brilliant 3-step investigative process. Step 1, gather massive amounts of genomic data from patients, specifically using genome-wide association studies, or GWAS. Step 2, mathematically predict or impute how those genes actually express themselves in the brain. And step 3, compare that disease profile against a massive database of how existing drugs affect cells, which is known as the Cmap database. The absolute genius of this process really lies in step 2, imputing brain gene expression using a powerful algorithm called MetaXcan. Why is this such a big deal? Because in psychiatry, you can't exactly take a biopsy of a living patient's brain, right? You usually have to rely on postmortem tissue from deceased patients. But there's a massive flaw there. Those patients were likely on psychiatric medications for decades, which artificially alters their brain's gene expression. So by using pure genetic DNA data from tens of thousands of people to mathematically predict the brain's baseline state, the researchers completely bypass the confounding effects of past medications. It gives us a pure, unpolluted look at the disease itself. Section 3, the reversal strategy, the lock and key. All right, so now that they had this pure genetic profile of the disease and a database of what drugs do to cells, they needed a way to actually match them up. The logic here is surprisingly simple, even though the math behind it is incredibly complex. Think of it like a lock and a key. If the genetic data shows that schizophrenia turns the volume up on a specific set of genes, the algorithm searches the drug database for an existing chemical that turns the volume down on those exact same genes. They are looking for reversed expression patterns. Basically, if the disease causes chaos, they look for the chemical that induces the exact mathematical opposite of that chaos. Section 4, surprising drug candidates, the lineup of suspects. The researchers ran this algorithm completely blind across 7 major psychiatric disorders, and the lineup of suspects it generated is just extraordinary. Now, the very first thing you'll notice is that the algorithm worked. Without any prior knowledge, it successfully identified known antipsychotics, like pimozidine, as top matches for schizophrenia. It even identified the famous SSRI fluoxetine for major depression. This validation is huge. It proves the math works. But the real excitement, the stuff that really turns heads, lies in the surprising new drug candidates. Let's look at how this plays out for specific disorders. Bipolar disorder and aspirin? No way, right? But yes, the algorithm flagged common non-steroidal anti-inflammatory drugs, or NSIDs, like aspirin and COX-2 inhibitors, as remarkably strong candidates for bipolar disorder. This is a massive hit because it aligns perfectly with this emerging medical theory called the neuroinflammatory hypothesis, which is the idea that mood disorders might actually be driven, in part, by inflammation in the brain. But hold on, it gets even wilder. For bipolar disorder, the algorithm also spit out simvastatin, which is a common cholesterol medication, and metformin, a widely prescribed diabetes drug. I mean, why in the world would a heart or blood sugar medication stabilize a mood disorder? Well, researchers have noted a shared cardiovascular pathophysiology in patients with bipolar disorder. In fact, separate clinical studies have already started hinting that statins might have a protective effect against depression. Millions of people take these pills daily, completely unaware they might possess hidden psychiatric benefits. And when the researchers pointed their algorithm at Alzheimer's disease, an area desperately starved for innovation, it flagged numerous common NSAIDs, as well as vasodilatory agents like naftridruferol, which helps improve blood flow. While clinical studies on these have shown somewhat inconsistent results in the past, the fact that a blind, purely mathematical genetic algorithm pointed right at them suggests there is a deep, fundamental biological link there that absolutely warrants rigorous, focused clinical study. Section 5, the future of repositioning, bypassing the bottleneck. What this Nature Neuroscience paper ultimately gives us is a completely new lens through which to view the pharmacy of the future. The advantages of this framework are just staggering. Because it's a hypothesis-free approach, it has the power to find drugs with entirely new mechanisms of action, finally getting us out of that 1950s rut. Furthermore, this computational matchmaking can be applied to literally any chemical with a measured expression profile. Think about traditional medicines, or even drugs that were shelved during trials for other diseases. They can all be run through this algorithm quickly and affordably. Now, we do need a quick reality check here to be clear about the limitations. You absolutely shouldn't go raiding your medicine cabinet to treat a mood disorder based on a computer algorithm. This paper represents the discovery phase. The researchers explicitly state that the absolute best approach for verifying these computational predictions must rest on careful, adequately sized preclinical and clinical trials. We have the lineup of suspects, now we just need the trials to prove their guilt. Or, in this case, their cure. But man, the implications are profoundly hopeful. For decades, the psychiatric drug pipeline has been agonizingly slow. This research suggests we might actually be able to hack the system by recycling the chemistry we already have. It leaves us with a pretty fascinating thought to ponder. What if the next major breakthrough in treating schizophrenia, depression, or Alzheimer's